First and foremost, I would like to extend my gratitude for watching my 5 minutes video. My name is Úrzula Franco-Enzástiga and I'm a postdoc at Dr. Price's lab. It is well established that women experience greater sensitivity to nociceptive pain, as well as heightened severity of chronic pain states. Some of these differences are driven by differential gene expression, which has been found specifically in human tibial nerve as well as in dorsal root ganglia in mice, but also in humans. Furthermore, it has also been shown that there are sex dimorphic mechanisms associated to pain, which have been shown mainly in preclinical models. The epigenome reflects the functional state of the genome by allowing certain DNA sequences or genes to be accessible for the binding of transcription factors or regulatory proteins, and therefore it affects the level of expression of some genes. Our work is the first of its kind to characterize the chromatin landscape in human dorsal root ganglia from organ donors without any history of chronic pain or neuropathy, and also to contrast the sex differences in this epigenomic landscape. With this aim in mind, we decided to implement an innovative tool that is called the Assay for Transposase Accessible Chromatin, also known as a TAXIC, that allow us to assay how the chromatin landscape looks in the cells, in this case, the human dorsal root ganglia. In our work, we use two different fashions of ataxic, specifically bulk ataxic and spatial ataxic. Spatial ataxic gave us the opportunity to resolve the chromatin landscape of different cell types. And for some of the experiments, we focused on neurons. We found that there's a sex dimorphic chromatin landscape in human DRG, and that some of the reads map to the X chromosome in females, which was not exactly expected. Moreover, we also observed that specific subsets of transcription factors were more abundant in males versus females. For instance, in males, transcription factors such as JUN and FOS, which belong to the AP transcription factor family, while in females, it was mainly EGR1, EGR3, and some SP transcription factors. From this, we validated that EGR1 was mainly expressed in females because we did RNA scope and we observed an increased amount of mRNAs in female DRG neurons. Among the genes that had increased accessibility in females, we found GABAergic, glutamatergic, and interferon-related genes, while in males, we found some channel-associated genes. Finally, since our ataxic data pointed to a heightened mapping of the ataxic reads to the X chromosome, we decided to assess cyst, which is a long non-coding RNA that compacts one of the X chromosomes in females. And what we found is that cyst is highly dispersed in the human dorsal root ganglia nuclei, specifically in neurons, in females, which was shocking because cyst is generally compacted in one spot, as it is observed in the other cell types in the human dorsal root ganglia, but the distribution was only observed in neurons. All of these differences that we observed at the epigenomic level between male and female could contribute to the heightened susceptibility to pain observed in females mainly.